Eukaryotes are a lot more complicated. So, gene regulation in eukaryotes. Okay, so let's look at the regulation that happens in just the nucleus to begin with. So one of the first ways that we can regulate gene expression is just based on whether or not we can have transcription occur. So we can regulate transcription. Anybody remember some ways that eukaryotes can have transcription regulated? We talked about like licking rats. You guys were laughing when I had a mom lick a baby rat. Yeah, I see Brandon like coiling and uncoiling. Okay. So this whole process is called chromatin modification. So chromatin is like unwound DNA, but a chromosome is when I take that same material and it's coiled into a tight package. So sometimes environmental cues can affect chromatin's coiling. So we can have DNA methylation, or we can have the removal of methyl groups, DNA demethylation, or DNA acetylation. So it's the addition or the removal of methyl groups or the addition of acetyl groups. So methylation will cause the chromosomes to condense. Demethylation will cause it to unwind. Acetylation will cause it to unwind as well. So if these methyl tags are added to DNA, it will coil and effectively silence the gene. That means that, like, sometimes you have identical twins, but they grow up in very different environments. And sometimes those environmental cues will methylate DNA. And so sometimes, even if you are genetically identical, you still have different regulation of genes based on the methylation or acetylation of the DNA. Because it's not enough to just have the gene, you have to have that gene turned on to express it. So that's one way that we can regulate transcription. The other way that we can regulate transcription is we have, when we talked about the stickleback fish, we talked about like the switches, or sometimes they're called enhancers. And for those, we'll have a section of the DNA where we have the gene. But upstream, like far away, we'll have the switch. And sometimes we call it the enhancer region, or we can call it the regulatory region, because enhancers, switches, they all regulate the expression of those genes. So we will have molecules, and based on what molecules are in the cell and available, they will bind to the enhancer region. And I have them in different colors because every single one of our cells has all of our DNA in it. But certain cells express some genes and others don't. Like my pancreas is currently making insulin, but my skin is not. They both have the gene for insulin, but just my pancreas is the only one that's producing it. So this is one way that we get cells to differentiate in their structure and their function, is by these enhancer regions. So once these are bound, then we will have the binding of mediator proteins, transcription factors, DNA binding proteins, and all of this, but they're going to give you a model of it, or they're going to describe it. But the main thing is, once we have these enhancers and they're bound to these activators, then we can have transcription factors bind. And these would be mediator proteins. And then here we have like transcription factors, which are other proteins. And then there's a promoter region right here where those are binding, just like we had in the bacteria. There's still a promoter. And once we have the mediator proteins binding and the transcription factors binding, then RNA polymerase will bind at their promoter, and we will go through transcription. So this is just regulating transcription either through chromatin modification or through the use of switches. Sometimes we will have a switch and it might activate multiple genes, because here it's activating this gene here, but you see how the DNA kind of bends over? Like it could bend over and activate this gene, and then it could bend the other way and activate a different gene somewhere else, because all of our related genes aren't necessarily close together on the same chromosome. So we're still in the nucleus. We're still talking about how eukaryotes regulate gene expression. We've just regulated transcription, which means that we had DNA, and now we have what we call primary transcript, or pre-messenger RNA. So the second way that we can regulate genes in eukaryotes is through RNA processing. And this happens in the nucleus. It's a lot easier for me to use a model that's already there. Here we have DNA, and it has this whole section is a gene, and you can see that there's introns and exons, and the whole thing is copied through transcription. And we have our primary transcript. In the cell, these introns are cut. And they're cut by these things called spliceosomes, and spliceosomes are made of RNA. They're RNA that acts like an enzyme. So they cut out the introns. What's left is the mature messenger RNA, and it's comprised only of exons. Yes? Yeah, so I'm glad that you talked about that, because we're going to talk about that next too. One thing that's important here is that sometimes you can have one gene produce different types of proteins. Because if I call these like different sections, like this would be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. There's 11 like sections there. And the only ones that are in the messenger RNA are the ones that are dark. 1, 3, 5, 7, 9, and 11. So the odd ones. But if we go through transcription again, different sections of the DNA could be tagged as introns or exons. Like, it doesn't have to be tagged that way all the time. So we could tag it different ways and push together new exons and make a whole different protein. So that's one thing, is we have the removal of introns. We put on a methylated cap, 5' methylated cap. That methyl cap allows it to squeeze out through a nuclear pore in the nucleus. And then at the other end, we have all of these A's, which are adenines, and it's at the 3' end, so it's a 3' poly A tail. So normally, your primary transcript is longer than your mature messenger RNA, even though we're adding a methyl cap and a poly A tail, because we have the removal of introns. That takes us through RNA processing. So now we're out of the nucleus and we're into the cytoplasm. We can now regulate transcription . There are enzymes that are going to degrade the messenger RNA. Those are called nucleases. It ends in ASE and it breaks down nucleic acids. So they're nucleases. Those nucleases will destroy the messenger RNA in the cytoplasm, but if we have a really long poly A tail, that helps the messenger RNA to last longer. So sometimes messenger RNA might last longer than others based on the length of its poly A tail. Because sometimes you only want to express a protein for a short amount of time, and other times you want to express it for a longer time. So that can regulate transcription. The other one that can regulate transcription, we can have the interfering RNA, or sometimes they're called microRNAs, because RNA is single-stranded. So if this right here is my messenger RNA, I could interfere with translation if I had small little pieces of complementary RNA that would make it double-stranded, because as soon as it's double-stranded, or even pieces of it are double-stranded, it's not going to fit into the ribosome properly, and it won't do transcription. So that's another way that we can regulate transcription. Then we can regulate the protein. The protein can be degraded by proteases. Those are enzymes that break down proteins. So sometimes you might make a protein, but it doesn't last very long in the cell because it's broken down by those enzymes. We also know that there is modification that can be done by the Golgi body. As well. And I think that's